Smile. 1, 2, 3. Okay, guys. Oh, that's a good picture.
Just drawing up the end.
You didn't do like the best show on too blending. I think it's perfect. You're almost as color as this.
I thought about it.
So you can
Exactly. That's what I wanted.
It's just one more interesting.
no way. That's amazing.
Yeah, one of my friends also, one of my friends also did a whole lot of things, theme thesis.
Really? But actually, it's also like just in the presentation, she was like almost on every chapter. She had like a small sentence that was referring to something. And she did like a reference to and all the stuff like that. And even one day of the jury actually noticed that like, okay, that's really cool. Really subtle, but really nice. See those people spending more time like including jokes in their presentation than actually work. Because we want to make it interesting.
I keep forgetting presentation.
That's all. I told him I'm waiting for Beyonce moment in the presentation. Something
It can be hard to convince to change things. You don't think so?
Wilt u gaan staan? U kunt wat naast nemen.
Hereby I open this academic ceremony in which Antonio Feliciano will defend the academic thesis, A Dance with Interactions, the Design of Dynamic Biomaterials to Influence Corneal Therapies. May I invite you to present a summary of your study and the conclusions of your thesis.
Thank you, Prorector, for the kind words. Dear family and friends, welcome to my PhD thesis presentation, A Dance with Interactions. So I think... All right, so at first I'd like to start by posing a question. When you look into the mirror, what do you see? Do you see a superstar? Do you see a scientist? Maybe you see a superstar scientist. After 6 years of research at Maastricht University, I'll tell you what I see. I see two perfectly curved transparent tissues, each a living optical lens responsible for 2/3 the refractive power of the eye. The cornea is our window to the world, and it is made up of 5 distinct layers. So from the outermost, which is the epithelium, to the center, which is the corneal stroma, which constitutes about 90% of the corneal thickness, and what we'll be focusing on here today, inside the endothelium. And despite being such a resilient tissue, tens of millions of people worldwide suffer from corneal blindness. And with only one tissue available for every 70 that need it, there is an unmet need for alternatives to the tissue supply. So current corneal therapies include transplantation, which is the gold standard. Typically can be done either with a partial or full thickness transplant. Otherwise, a patient can elect for a keratoprosthesis in which an acrylic glass is embedded into the cornea. However, this is associated with high risks, such as chronic inflammation and risk of rejection. So many biomaterials have been developed to try to solve this problem, but none have yet been approved. So materials like collagen, PEG, and also gelatin, as well as decellularized cornea. However, we still have problems with these materials. For example, these materials are poorly integrative, which can cause corneal melting or corneal necrosis, and also cause a loss in optical clarity. So let's dive in a little deeper on how we can solve these problems. So first, poorly permissive materials are not good for integrating with the body and with the host tissue because if a material network is too dense or doesn't allow cells to infiltrate, the ACM can no longer be reproduced inside the gel matrix or material matrix. So here's a good example of a corneal inlay, which is made up of a PVDF polymer. Because it doesn't allow tissues to infiltrate, they just start to encapsulate the material and form sort of this scar tissue or fibrosis on the surface. However, when we build integrative materials, permissive materials, cells can infiltrate and start to reproduce extracellular matrix, giving better and improved tissue integration. Now, the second challenge is this main cell type is the corneal keratocyte, and it's very sensitive and can phenotypically drift to a fibroblast or a scarring phenotype. So it's important that we do not disturb this cell, as it could cause a loss in vision over time. So while you might start with a very transparent material, as a patient is living, it becomes less clear for them. So we aim to solve these problems with supramolecular and non-covalent interactions that mimic interactions found biologically. For example, in the cornea, collagen fibers secreted by corneal keratocytes are held together through supramolecular interactions. We can use these interactions because they are reversible and can associate and dissociate. And this is what is happening inside of our body. We can also use discrete supramolecular units, such as host-guest, UPs, and electrostatic interactions to try to build materials with these interactions that are biomimetic. Additionally, we can engineer them using this association constant to build stronger materials and also maybe weaker materials to allow permissivity in those scaffolds. So in this thesis, we looked at 4 subsets of supramolecular or non-covalent chemistry. We also designed each of these for a particular corneal tissue engineering application, including corneal-resistant implants, in vitro models to recapitulate corneal tissue dynamics, drug-eluting contact lenses for wound healing, and a microgel scaffold for corneal tissue regeneration. So in chapter 3, we'll start with the zwitterionic supramolecular copolymer. So here we wanted to mitigate the foreign body reaction, and one of the essential steps in preventing that is preventing protein absorption and cellular adhesion to a material. So here we developed a copolymer which could be anchored using supramolecular groups like UPs to the surface of a UP functionalized elastomer. These zwitterionic moieties then act as cell repellents and also preventing BSA from absorbing, but also other proteins. And this gave us the ability to modularly add supramolecular groups to increase coating longevity. So here we see that we, with 5% UPs in the backbone of the copolymer, we had better surface coverage with our copolymers. Additionally, we were repelling BSA from the surface of these, so it was protein resistant. We also did cell studies to see if this antifouling effect was also with cells, and we found that there was a decreased amount of cell adhesion when we applied these coatings. So this could be used potentially to mitigate foreign body reaction, but more study is needed. In the next chapter, we looked at recapitulating corneal tissue dynamics. So it's not well studied in literature, but the cornea, like other tissues, exhibit this time-dependent mechanical property, like stress relaxation. So we probed the surface using a nano-indenter, in which a indenter puts a constant strain on the sample, and we found that over time, stress was dissipated, resulting in a stress relaxation. We then tried to modulate the stress relaxation in an in vitro model. This was done using work inspired by Chad Urey's group, in which PEG is coupled to an alginate, resulting in very fast relaxing gels when combined with calcium. And as we add UV, so UV dose, in the presence of a photoinitiator, we can go from a very fast relaxing environment to a much slower relaxing environment. Then we can plot this and see how it compares to the decellularized cornea, and we can see that we can move from a very fast relaxing environment to a slower relaxing environment, maybe getting closer to a decellularized cornea, more closer to the in vivo situation. As we grew cells on these constructs for 7 days, we found some interesting cell behavior. So first, we found there was more philopodia, or more sensing of these cells in this environment when they were on slow relaxing gels as compared to fast relaxing. We also found that alpha smooth muscle actin, which is a fibroblast marker, was downregulated. And there was an improved transparency in the constructs when we looked at them under transmission. So it seems to us that there is this propensity for these cells maybe to prefer the slow relaxing environments as opposed to the fast relaxing. In the next chapter, we wanted to fabricate drug-eluting contact lenses. So these are kind of a schematic on how these cells change when a wound healing event occurs. So they're normally a quiescent cell that starts to become more fibroblastic and even myofibroblastic, and these secrete scar tissue. And the idea was, if there's a break in the stroma, can we have a contact lens hydrogel deliver an active ingredient to prevent this phenotypic drift? And what we found was in literature that there was some propensity for vitamin A or retinol to be used with corneal keratocytes to improve their phenotype, but also improve their the stromal matrix that they were producing. So interestingly, vitamin A or retinol can be encapsulated in a higher order stoichiometry with cyclodextrins. So inside the two cyclodextrins is one retinol molecule, and we wanted to verify this using a phase solubility plot. We found that as we increase the amount of cyclodextrin, we sort of reach a plateau with how much retinol we can encapsulate. And even at some point, we get a drop in the amount of retinol that's being encapsulated. So this is indicative of maybe a higher order stoichiometry that's maybe aggregating and falling out of solution. We then hypothesized that this could maybe yield a supramolecular hydrogel, as adamantane and cyclodextrin have been used before for these same types of gels. So we then loaded retinol into increasing amounts with this alginate functionalized with cyclodextrin. And we found when we loaded it 4 times the amount of retinol and cyclodextrin, there was a gel-like behavior in the gels with retinol as opposed to without. So there was some sort of change in the structure of these materials, and it could be due to this encapsulation effect. Also, we found that there was a self-healing. As we exposed the material to very high shear, it became more liquid-like and then reverted back to a gel state when we had low strain. However, when we tried to use these under physiological conditions, at 37 degrees Celsius, for example, they completely fell apart in cell culture media, so we wanted to try a new approach to make these more stable. And one way you can do that was trying to do a different approach like templating or molecular imprinting, in which you solubilize the retinol in 2 cyclodextrins that have been functionalized with thiol groups. And then with an alginate norbornene, we tried to perform a thiolene click chemistry where alginate could be cross-linked using this dynamic bond. We found that as we changed the ratio of cyclodextrin to retinol, that material properties were changing as well. So with the 1-to-1, we see a more liquid-like material. It's also more opaque versus the 2-to-1. We also found that these were more robust, so they could withhold a shape after being molded. We also found that there was a different dose release profile with each of these gels. And when we were growing corneal keratocytes in the presence of these, there was a reduction in large myofibroblastic-like cells. So we went from a very large cell in the control to smaller cells that were also a bit more dendritic. And then we also found that there was this therapeutic window. So at 2 high concentrations of retinol, we had a cytotoxic effect, but then at lower concentrations, we saw a halting of proliferation, which might be what we want with corneal keratocytes, as they should be more in a quiescent state. Okay, in the last chapter, we wanted to create microtissue in the lab using a polyrotaxane motif. So here we developed polyrotaxanes using alpha-cyclodextrin and PEG as the axle polymer. Here we capped with a 4-arm PEG norbornene, and we found that increasing amounts of alpha-cyclodextrin, you can modulate material properties, and also at 15% we can see a self-healing effect when we dice these. However, these materials tend to be, these bulk materials are not very cell integrative, and so we chose to do a granular polyrotaxane microgel scaffold, which would enable cells to infiltrate. So here we ran with a microfluidic chip, microgels around the order of 100 microns in size, and these were able to pass through a needle to be injectable or 3D printable. Also, these gels exhibited an interesting thermoresponsive behavior where they started to become more adhesive with temperature, and we were able to create a cohesive corneal-shaped scaffold that we could grow cells with. So after a couple of days in cell culture, we saw great cellular infiltration, which we did not see on bulk materials, and we saw sort of a multicellular layered cellular organization. So perhaps these gels are good for helping organize the cells in a way other than TCP, which was just giving us cell aggregates. So with that, I hope I've shown you that we can use this dance of interactions to not only enhance anti-fouling coating longevity, but also recapitulate viscoelastic properties. We can also observe less myofibroblastic-like cells with retinol. And we have scaffolds capable of cellular infiltration and cellular organization. So next time you look in the mirror, I hope you see what I see, which are 2 dynamic, resilient, and vital tissues. And know that we're just one step closer in helping corneal blind people see again. Thank you.
Thank you for your presentation. The opposition will be now opened by Professor Nuits, Professor of Ophthalmology at the University of Maastricht. Professor Nuits was chair of the assessment committee, and he is the secretary of this degree committee.
Thank you very much. Mr. Candidate, I first would like to congratulate you and also your promotion team, the 2 promoters and the co-promoter, with this very nice thesis. It was indeed very interesting to see your dance with interactions using supramolecular chemistry, and I think you have experienced how complex the cornea as a human tissue is also in that. It was a pleasure to read it, although some of the chemistry specifics go beyond my expertise as a clinician primarily, I must say. And it is also great to see, I think that this is the first PhD thesis to originate from the EyeSight research project. So it's the money of the Insight funding agency is well spent, I would say. Let's go to chapter 3. In chapter 3, you describe the anti-fouling technology to enhance the biocompatibility of intracorneal inlays that we use to correct refractive errors in the cornea, as you know, and to reshape the cornea. Now, my experience with corneal inlays is quite diverse, I would say. As you know, there is, at the moment, no commercial implant on the market anymore. When I did my research fellowship in '93 in Atlanta at Emory University, there had been 2 Dutch colleagues, ophthalmologists, predecessors, who studied corneal hydrogel implants actually in monkeys there. And it worked fine in the beginning. But after some time, they saw haze and all kinds of problems. In '97, I used a new treatment, actually, polyurethane, that was used to close dura mater defects, actually, in the cornea to close corneal defects. And that's white. So if you look at it, it looks actually awful. But it did the job, it closed the defect, and we saw that after 6 months to a year, cells were going in and became more or less a little bit transparent. So we have 2 mechanisms here. One is looking for a polymer that can go in there where there is no tissue interaction, and the other one is actually, creating a scaffold where the only cells, the own cells can come in. Probably for the future, some stem cells could get in there. And you are working now at anti-fouling of a polymer. So do you believe that anti-fouling is really going to do the job, in the sense that we won't see these side effects that we see in terms of corneal haze formation, fibrosis at the end?
Thank you, dear highly esteemed opponent, for a great question, especially as it relates to putting these anti-fouling coatings into a clinical setting. I think when we designed the anti-fouling coatings, we were trying to prevent cell adhesion, protein absorption at the onset of implantation. And I think this is a very sort of chaotic environment when you're implanting. So there's a lot of potential for adding more proteins or cell debris, perhaps, during implantation. So I think the way our coatings seem to be working is that it would prevent sort of this initial adhesion phase. So primarily during the inflammation phase, you probably have a lot more cells, maybe that are activated. And so by preventing those cells from seeing the material with the anti-fouling coating, I think maybe we could prevent this corneal haze in the future. But then it becomes also a matter of how do cells then infiltrate or work with the material after the coating is gone. And so I think that's something that would need to be studied further as well.
Now, I saw in your experiments that you did the cell adhesion, of course, and you studied that at 24 and 48 hours.
Yep.
Now, our experience clinically is that the wound healing takes a little bit longer than 48 hours. So
Sure.
Just to put it in perspective, why did you stop your experiment at 48 hours? Why didn't you set up an experiment that could go, you can keep that in culture for a couple of weeks or even longer if you wish.
Yeah.
So why did you stop that at 48 hours?
I think, as a young student, I saw, okay, there's the cells starting to grow already now after 48 hours. So I was a little worried about the applicability, so going further in days. But I think there's also, we have to think, we seeded these with a lot more cells than maybe we were that would see in the in vivo situation. We also challenged the coatings with being in a BSA overnight. So I think these are ways to challenge the coating and try to make it not work. And so maybe it's not clear how those challenges relate to the in vivo situation, because you wouldn't necessarily have it floating in a BSA solution or FBS, I'm sorry. But it would be, I think, good also to perform those experiments longer and see how long it lasts.
So before going into animal experiments, would you be happy with these results, or would you do some additional experiments before you go into further animal studies?
Yeah, I think...
because that's what we really need before we can go further, of course.
Right. We need some good data to suggest that this would be good in animals. And I think, we did challenge it quite a bit. And I think having challenged it, and we did still see some good anti-fouling, at least within 24 hours. I think I might feel comfortable seeing it in an animal, especially because the materials are biocompatible. the elastomer that we were using was specifically tailored for this kind of application. So I think I wouldn't be too afraid of like negative outcomes.
Okay, so we will see in the future if that's going to happen. I have time for another question, I think. Yes, so I would like to go with you to chapter 6, where you study actually or describe the use of granular polyurethoxane hydrogels for tissue regenerations. Yeah. So we have many unmet needs, but one unmet need in my field is, you know, those patients who got a piece of iron in their cornea, and it's exactly in the pupil size, and they get bothered by, you know, the scar tissue, yeah, and the glare and the halos that are being related to that optical phenomena. Now, we have excellent lasers in ophthalmology who can cut out these scars with micrometer precision, actually. But then we have to put something back there, yeah, and usually you have to suture it, and then it becomes a mess. And the patient is actually worse off with these very small lesions of 1.5 to 2 millimeters than it was before. And we cannot put a big transplant in there. That makes no sense. So these solutions, have they the opportunity to become adhesive, like we have tissue glue and all kind of stuff that we use in ophthalmology, to become adhesive? Can you fill that? Can you modulate them that they could do the job in these cases? And what is there? You said something, there was one graph about the optical transparency, but I have no information on refractive index and that kind of stuff. Right. So what is the future for the clinical applicability of that?
Yeah, thank you for the question. As it relates to the clinical applicability of the granular polyurethoxanes, I think we have to be careful using those specifically in a point where a patient needs to see right away. These gels tend to be opaque because of the polyurethoxanes in the polymer structure, but we did show that they could be degraded over time, then increasing transparency. So I think when we look at the transparency results compared to the human cornea with this potential corneal-like material that the cells are organizing into, there's still microgels present, so we would have to find a way to either remove those gels or wait for them to degrade. Mm-hmm. So maybe there's like a degradation step that we do to the material. But that being said, these microgels do, when we expose them to higher temperatures, they adhere really well to one another, and they actually do have a really nice adhesion property. So we would have to sort of, with those limitations of having an opaque material, I think when we anneal the the granular polyurethoxane material, it could be useful as a way to like fill a void, for example. Maybe not necessarily. Yeah.
The final small question then. Could these gels be applicable also to fulfill our dream, and that is in keratoconus, the keratoconus disease, mm-hmm, to fill the defects that we have in the tissue, and then to mold it gradually up until we have the shape that we desire? Is that a possibility? Yeah.
I think what's really, what I really love about the granular materials is that they're these very small microgels that can fit a shape really well. And then through the interstitial voids, then nutrient flow, cells can move through it. It's very good in that way, that it's not preventing, whereas I think some of our inlays would prevent a nutrient flow and then cause problems later on. So I think in that respect, I think they're really good, but we have to figure out how to use them with this opaque, and maybe a different chemistry could even be used.
Okay, we're looking forward to the future results of this. Thank you very much for the question. I give the word back to the director.
Thank you. The opposition will be continued by Dr. Bolognien, Assistant Professor at the Maastricht University. Dr. Bolognien was also member of the assessment committee.
Thank you, Prorector. Mr. Candidate, first of all, my congratulations to you and all the supervising team for this excellent work. I really liked how the thesis was organized, very clear, very interesting finding, and with an eye we can really say, toward market application, which is not always a given, and I particularly enjoy this part. And maybe I'll start with the final chapter, 7, where you discuss these entrepreneurial activities and commercialization potential. And you mentioned microgel platforms for tissue cultivation and also the possibility of synthesizing cornea. Mm-hmm. But I'm wondering, what were the aspects you did not consider as a scientist which became really relevant when you talked to the private sectors?
Dear highly esteemed opponent, thank you for the very important question. So when we look at the polyrotaxane materials, you're asking what maybe I missed my first go through, and then what maybe I've learned since. So I think when it comes to these polyrotaxane structures, I immediately when we exhibited a so we added temperature to the scaffolds. We saw that there was a stiffening effect. This also resulted in a decrease in the microparticle or the microgel size. There was this idea that supramolecular materials should be reversible. Mm-hmm. And when I saw that the material was not reversible, I think I was a little upset with myself because I don't know what I made, right? But these cyclodextrins, I think on the polyrotaxane motif, what happens is they start to aggregate with one another, and they form more of like a crystalline structure that when you drop temperature, they don't really come apart. So I'm not sure too much on the mechanism of why that is. Others have used sort of like a methyl group to prevent this crystallization from occurring. But I think that's something that, despite seeing that, I was a little thrown off by, but I think we used it in the end as leverage, so.
Thank you. And if we talk about longevity, was this a point that the private sector brought up? And I tied in this question to chapter 3, where you mentioned a bit about the longevity of these copolymers. We heard from the previous opponent that you tested up to 48 hours in vitro. Was that also because the hydrogels are not stable after that, or this is not a concern?
So the materials that we were using for the zwitterionic copolymers, we were applying to a UP functionalized elastomer. So these have the elastomer itself is quite stable in that it has a very good longevity. The coatings, however, those were a little bit on the weaker side, and I think what happens is in one of my propositions, I say, water giveth and water also taketh away. So we would apply these coatings, and as we applied water or, cell culture media, we started to notice that maybe the properties were starting to be less or reduced. And originally I was kind of looking at, okay, maybe we should do like a covalent grafting approach to improve the longevity. But I think this might be a good example of using supramolecular, chemistry to have a temporal relationship with our materials.
Mm-hmm. And if we could apply the modifications you just mentioned, just out of curiosity, how long do you think we could extend the use of these copolymers? Are we talking about days or?
Yeah, actually, another limitation about the chemistry that we explored was we had a very specific co-solvent system to do the reaction, which only let us get up to about 5 mol% in UP on the backbone. So more UPs on the backbone could potentially offer better longevity because there's more chance for them to interact with one another, but also it's more hydrophobic, and then you maybe have these hydrophobic areas on the elastomer and then the zwitterionic on the surface. So I think there is definitely a chance to improve the coating longevity just through molecular increasing the modularity of the UP group on the copolymer backbone.
Thank you. Very nice answer. Then I have a question on chapter 4. You observed when you tested the medium and slower relaxing hydrogel that you have more philopodia from the cells. And in other parts of the same chapter, you say that these gels are more physiological because keratocytes should be relatively immobile. Mm-hmm. So how do you match these two observations? From one hand, filopodia are appearing when cells are migrating, mm-hmm. but on the other hand, you are saying the cells are more immobile and quiescent, so to say.
Yeah, thank you for the very important question, especially when we look at the keratocyte phenotype. A lot of times we look at the morphology of these cells and how they interact with their environment. I think when we looked at the cells with the slower relaxing gels, they had these more protrusions. Under the microscope, they were just kind of feeling the environment. I don't think they were necessarily too mobile. They weren't moving too much. On TCP, they were very active, so we could compare sort of how active they were. On the faster relaxing hydrogels, the cells didn't have these sort of like fingers, feeling or sensing. It was kind of more of a circular edge than a dendritic morphology.
Thank you. And then the very last question. In chapter 4, you showed also higher DNA content on hydrogels compared to standard tissue culture plastic. And you suggest that these 3D constructs could have provided more volume for cells to grow. Do you think is really the point that simply the cells have more space to grow, or is the hydrogel influencing some signaling pathways and cells are proliferating more, or what's happening there, in your opinion?
Yeah, I think that's a very important point. When we were initially imaging the gels, so when we started, we seeded the gels on top of these hydrogels. And when we kind of looked on top, we noticed that a lot of the cells had started to go inside. And so trying to capture these images on the microscope were a little bit more difficult. And so we were wondering if maybe they're starting to migrate into some of these scaffolds, or maybe they fell in, I'm not exactly sure. So I think making that statement, we felt comfortable saying, okay, there seems to be more than just a 2D surface with cells. They seem to also be embedded within. But that being said, that's not a clear answer for that. So I think more study would have to be needed to say that conclusion.
Thank you. Then I'm really satisfied with the answers, and I give the word back to the prorector. Thank you.
Thank you. The opposition will be continued by Professor Dankers, Professor Supramolecular Biomaterials at Eindhoven University of Technology. Professor Dankers was also a member of the assessment committee, and we highly appreciate your participation to this academic ceremony.
Okay, thank you very much, Mr. Chairman. Geachte promovendus, first of all, of course, I would like to congratulate you with your beautiful work, and I was also very pleased that we at least could collaborate a bit within the eyesight project. Also would like to congratulate your promotion team for this nice achievement. Of course, I still have some questions, and when I start at the cover of your thesis, I see a little bit of a scary eye, which I can maybe understand why that is, and you want to dance. So my question is, whose eye is this? What is it looking at, and who are you going to dance with?
Well, it's looking at you.
Okay.
No, just kidding.
No, okay.
It's inspired by Game of Thrones, yeah. So it's a dance with dragons. The inspiration kind of comes from just looking at my thesis generally, okay. Where each chapter I felt like was kind of like a Game of Thrones, where every supramolecular chemistry I would get excited about. I'm like, that's the one. That's okay. The one I'm going to work with. And then I would see its like limitations, and I would try another one, and then I'd be like, this is the one, okay. And then tried another one. So there was, I think, 4 different supramolecular chemistries here.
Okay. And which one would you now choose then for your implant? If you would help colleague Professor Nuits.
Which one would I choose? All of them, no.
No, you can only choose one.
Yeah, I think we saw the most interesting effects with the microgels, and that's just because of the granularity. Okay. It could be a different chemistry as well to modulate.
So that's the route we should go. Okay, that's clear. But then the dance, who are you going to dance with?
Yeah. So I'll dance later tonight.
Okay, very good. But you don't want to share with whom? Or do I need to look that? But that's fine. No, good. Then let's go to the propositions. I would like to go to proposition, I have to check. 3, maybe the second can read that one.
Number 3, correct?
Yes.
Faster stress relaxation is not always better. Biological systems require balance between stability and adaptability.
Okay, thank you very much. Where do you see this is not better in your thesis?
Yep. So thank you, highly esteemed opponent, for that question. I think when we look at literature, typically a fast relaxing hydrogel has been shown to increase spreading, proliferation, can also trigger differentiation. And I kind of went into this specific chapter thinking that maybe that's not always the case. Maybe you need a slower relaxing material.
And if you would go to your thesis?
Yeah, I think that the slower relaxing material might be better for in vivo like corneal keratocyte function.
Okay, but I would like to have a little bit more detail. What is slower? Which time scales are we speaking about?
Yeah, so in our experiments, we looked primarily at the between 1 and 10 seconds. So this is sort of the time scale where cells can maybe interact with the environment and sense the material properties.
Okay.
So on this time scale, we saw that very fast. Sorry.
Okay, but what is slow then? 1 second? 10 seconds? Half second?
Yeah, it's around 5 seconds for the tau.
And fast?
Yeah.
What is fast?
And fast is around 1 second, I think.
Okay.
Yeah, so it's the span of range for relaxation in this regime is very maybe small, and so another system might be better at deciphering out what those a longer relaxation time.
Okay, but if you now go to your thesis, chapter 4, you also discussed this with the previous opponents. You show that the formation of these protrusions is that now in the what is now fast and what is slow? Can you comment on the time scale versus why the cells would make these protrusions or not?
Yeah. So on the slower relaxing gel, so this is like with a tau of I think 5 seconds, and this we think is maybe closer to what the in vivo situation is like with the cornea. The cornea also exhibited stress relaxation, but it was also not very fast.
And they make these protrusions, isn't it? That's what you show.
On the slow relaxing.
Yeah, but why?
Yeah, I think, and this is just a hypothesis, so I think maybe if there's like a wound healing event or if there's something that maybe triggers the cornea to deteriorate, this may exhibit very fast relaxing behavior. So maybe the cells are picking up the fact that, okay, so a fast relaxing environment is something where I need to heal. And so I need to create...
because if you look to the healthy cornea, I also don't know the answer here, but then isn't then what you see in your slow gel better? I'm not sure. You have to give me your opinion. But the one with the slow relaxing gel seems to have more healthy cells, or is that not correct? What I see?
Yeah, correct. So we saw that the cells were circular with the very fast relaxing, but then with the slow, they were more dendritic.
So do we really need to have this dendritic structures? Because I recently, why? I still don't have that answer.
Yeah, so I think when we have these cells in this morphology, they may have less reason to, or they may have less alpha smooth muscle actin production, which is a fibroblastic marker.
Okay. Is that what you see or not? Did you stain them?
We did stain them, and we didn't see any alpha smooth muscle actin in the gels. But that could also be, in that way.
Okay, yeah, because I was doubting whether it was not a matter of that they can't really adhere on your gels, because you use this linear RGD, which in our hands never works, but that might be also, I don't say it doesn't work with you, but might that be also something that could have influence on your adhesive properties of these cells?
Yeah, I think so as well. I think we were expecting to see a lot more cells than we saw.
Okay.
So maybe there is this potential for this problem with cell adhesion.
Okay, and what was the concentration of your IgD? Do you know that?
750 micromolar.
Okay, that's micromolar. Yeah, okay. Okay, it might be a bit low, but... Okay, good. Then I have one more question, if I'm allowed, Mr. Chairman. I also, of course, love your chemistry. So I would like to go to chapter 3, and there you do a reaction of the UP isocyanate with your HEMA group. And then you have a nice NMR, and then you see some double peaks of the double bond, the proteins at the double bond versus the alkylidine protein on your pyrimidinone rings.
Right.
Can you comment a bit on that? We can go to that picture, but I have to see where it is. It might be figure 2A, page 62. I'm not sure. Let me see. Yes, a bit tiny, but there's 62.
Yep.
So there you see the NMR. Let's go to A, figure A.
Yep.
And then if you take the zoom, you see there a D and F.
Yep.
And an E. So can you comment a little bit on the D and F?
Yeah, so the D and the F are the acrylate protons. So this is telling us that we have the double bonds available there, so we could do the raft polymerization.
Yeah, but I see 2 peaks for A and for F. You don't see that? I see that.
Oh, E.
No.
A? Sorry.
It's tiny. I have to read. My eyes are not so good anymore. It's, let's say, in figure 2A, and then the zoom of A. Then you see for D and F.
Yeah.
Yeah. Protons D and F. You see 2 peaks. What is that? Yeah.
Yeah. There's also a smaller peak, I think, with E2, maybe?
Yeah, but I come to that later.
Okay.
But first these 2.
Okay. Wow, it's been a long time since I've looked at the spectra, to be honest.
Okay.
But
might that be your unreacted double bond, or you think you don't see that?
Maybe it wasn't purified well enough.
Okay. Yeah, because if you would go to E, 2A and then proton E, to make it clear, you see also normally,
small,
what could that be? So
small hump there.
Yeah, I might even see 1, 2, 3 peaks for proton E.
Yeah.
So do you have an idea what that could be?
Yeah, maybe it could also be maybe some of the reactant still showing up, but...
Or did you have a look whether you have a keto or enol? Pyrimidinone versus pyrimidinol?
I did not look into that, no.
Okay, because that might be...
that's a good point, yeah.
Okay, good. Then I think I'm very happy with all the answers, and give the floor back to the chairman. Thank you very much.
The opposition will be continued by Professor Weber, Professor Chemical and Biomolecular Engineering at the University of Notre Dame in Indiana, US. Professor Weber was also a member of the assessment committee, and thank you also for coming over for this academic ceremony.
Thank you. Happy to be here. So, dear candidate, congratulations to you and your team on, I guess, what is eye-catching work here. I want to start with a conversation about Proposition 6. I'm happy to read it, or it can be read for me.
Biology is the study of supramolecular interactions that sustain life.
I'd like you to defend that proposition.
All right, yeah. So I think, dear highly esteemed opponent, thank you for the great question. When I look at biology, I'm always fascinated by what the body's doing and how it can just do what it's doing. A lot of these interactions that take place are not necessarily covalent interactions, because then that would be too elastic for us to move. We have the ability to see, to dance with interactions. And I think this is because of these supramolecular interactions. If we were made completely out of rigid material, we wouldn't be able to be dynamic in that way. And I think when we look at the cornea specifically, these supramolecular interactions give the strength to the cornea, but also the transparency. So I'm just constantly surprised that biology can use these kinds of interactions and get away with it and last for so long. You know, we age, I don't know, we could age to 100 years old, but some of the interactions that I've worked with are so transient they dissolve in a few seconds. So when I look at biology and its mastery of supramolecular chemistry, I am in awe.
Okay, so when you defined supramolecular chemistry in some of your introductory work, and in I think chapter 2,
Yep.
you define a parameter KEQ. So what is KEQ?
So KEQ is made up of the association and dissociation, sort of a ratio between the two. So how well you can engineer materials' strength, for example, can be used by increasing the association.
Okay, maybe more specifically, what does the EQ stand for?
The equilibrium.
Equilibrium, okay.
Yeah.
Is biology an equilibrium system or a non-equilibrium system?
Yeah. I think, you know, to be fair, things are constantly changing and moving in our bodies, and somehow it manages to sustain itself. So I'm not sure if that's, I feel like that is chemistry in its fundamental form. So
I would posit that human life only reaches equilibrium long after death, after decomposition is basically completed, right? And so I think we are inherently non-equilibrium in life, right? And so I wonder how that plays into this idea that biology is merely the study of supramolecular interactions, if supramolecular interactions are defined by their equilibrium states.
Right. That's a good point. Yep.
Cool. Okay, great, thank you. I wanted to go to chapter 4 and ask you to speak about the origins of stress relaxation in these alginate hydrogel systems, first off.
Yeah. So originally, this work was inspired by Chaduri's lab, which coupled very large and small molecular weight pegs to alginate molecules. And then after cross-linking with calcium, they found that very large molecular weight pegs were able to exhibit fast relaxing behavior.
Where's the relaxation coming from in that system?
I think that's an important question. I believe that there might be some interference by having these very large molecular weight polymers that are then competing or even moving calcium bridge points, and then maybe, I don't know, pushing them to come apart. I don't know if that's exactly the phenomenon or if we have described that with these gels.
Yeah, so alginate is primarily an ionic gel, right? And so you get the egg box structure of the alginates coordinating around calcium or barium or whatever your divalent ion is. Is that the dynamic piece in a typical alginate hydrogel? Before you start modifying it, is that where the bond dynamics are originating, do you think?
I think it's a very stable bond, the electrostatic interaction with calcium. So, but they can come apart. So I think there is a reversibility in that bond. I think that's how these gels were manufactured and made to give them this stress relaxing property. So I don't know where else necessarily would give that same reversibility.
Okay. And then you use UV time as the mechanism for reducing the stress relaxation rate of the material. I presumably to increase the formation of kinetic chains among your pendant norbornene groups. Why do time as opposed to, say, altering the degree of substitution of the norbornene?
For this particular study, time was convenience, so we could easily take the polymer that we had that was already functionalized with the alginate norbornene. So it had a specific amount of norbornenes already, and then we could just use the same batch to make different gels. If we tried to increase the amount of norbornenes or the amount of functionalization that's presented on the polymers, I think it would require a bit more work to prepare those materials. And so just studying time was a good way to use the same batch and then quantify its stress relaxation after exposure.
Okay. And then I guess I wanted to get lastly to the method of quantifying stress relaxation. And so you use nanoindentation to determine the stress relaxation of the corneal tissue.
Correct.
And then you use a rheometer to calculate the stress relaxation of your gels. Do you envision any mismatch in length scales between these two measurements? And you're kind of trying to push towards this limit of what you say is the stress relaxation of the cornea, but you're using a different experimental technique to do that. It seems to me that there could be some length scale mismatch between an AFM tip and say a bulk rheometer in measuring these kinds of things. Do you envision any of that in practice?
Yeah, I think that's a great point. Using these two different tools we can get stress relaxation properties, I think, a bit easier with our alginate gels. When we looked at the rheometer, it's easy to set up, it's easy to run. With a nanoindenter tip, it's a little bit more finicky. Sometimes the gels are very sticky, and then you can easily break a tip. So just from a ease of use and handleability, we use that approach to experiment with the rheometer and then also experiment with the nanoindenter. That being said, we also verified with the nanoindenter the slow and fast relaxing hydrogels. I believe it's in like a supplementary figure where we also saw that there was a propensity for UV light exposure to make the hydrogel more elastic behaving than the fast relaxing. So there is a trend. How those two are coupled, so the rheometer and the nanoindenter, I don't have a necessarily clear answer on how that works, or how that would be relative to one another. But, we did try to validate that with the nanoindentation.
You do plot them on the same axes, and so that's why I was curious if you think they are actually, similar measurements. I think with that, I can just close by, again, congratulating you, I guess, also on your perseverance to get to this point. I know it's 17 years or so since you were an undergraduate working with me at Northwestern, and so to see you from that point till now, I guess we both have a little more gray hair and we're a little older now, but it's fantastic to see, and congratulations on your perseverance. I'm satisfied with your answers, and I'll yield the balance of the time.
Okay, thank you. The opposition will be continued by Dr. Speidel, affiliated researchers at the Karolinska Institute in Sweden. Dr. Speidel was also, we also thank you for coming over for this academic ceremony.
My pleasure. Thank you so much. And thank you, Mr. Candidate, for a very nice thesis and for a nice presentation this morning. It's a real pleasure to be here and to ask some questions. So to start off, it was a real joy to read through this thesis and also see the evolution of your scientific evaluative capabilities. I wanted to ask you about so you used a number of different methods for measuring cell viability, metabolic activity, and I was curious if you were to revisit the viability experiments that you conducted in Chapter 3, given what you've learned later on and that you've shown in later chapters. Would there be any changes that you would make in those experiments, and if so, what?
Yeah. Thank you so much, our dear highly esteemed opponent, for the question, especially as it relates to my progression as a scientist and the viability of these assays, or the viability assays. I think as we, there was actually a struggle throughout the entire PhD journey with viability assays, specifically MTT assays, so the metabolic activity assays. As we were trying to use or trying to make a corneal keratocyte that I guess would be quiescent, when we were running MTT assays, we didn't have a lot of it almost looked like they were not viable, but I'm not sure if that's because maybe they were just less metabolically active and more in their quiescent state. But I think that's something that we could explore more. But I think as we did live dead imaging in the end, it's a really good way to see how the cells are alive and which cells are dead compared to maybe an assay like Presto Blue, where we don't necessarily see that. So I think there's limitations and challenges with each one, and then there's also benefits for each.
Yeah, great. In that reply, you mentioned some of the complexity with these types of assays because you have to capture both cell number and then also some sort of metabolic activity measure. Yeah. In light of that, is there any adaptation or addition that you might put into the assay further?
Thank you. So I think we're trying to understand how much more viability information we could use to make sure that our materials are biocompatible, correct?
Yeah, I guess, so you touched on one of the kind of cruxes of the issue, right? So you could have, if you have the same number of cells in different materials, yeah, they may still not have the same level of metabolic activity, correct?
Yeah.
Yeah, so if you were to try to assess this, how would you do that?
Okay, that's a great question. I think we saw some of having the visual analysis, so with the live dead, also really helped us see what's going on. So for example, with our retinol gels, we first probed this with MTT assay, where we saw very low metabolic activity, but then when we tried with live dead, we saw there were still cells there, so we didn't really understand. So I think maybe there's multiple of these assays that you have to screen, especially when we're working with a quiescent phenotype like the corneal keratocyte, just because we can't necessarily make those conclusions without having more data.
Yeah, okay, sounds good. Great. And then a number of the different chapters, you present materials that use UV cross-linking mechanisms in order to generate the materials in their final formulation. Given, and then there's also the retinol that you creatively use as the cross-linking mechanism in one of your materials. I was curious how you reflected on if you were to apply these materials in humans or even animals, what the impact might be, given that they would be in the eye and exposed to UV over time, what your thoughts were around that.
Yeah, thank you so much for the question. We think, so when we were developing the stress relaxing hydrogels, we envisioned this as primarily an in vitro model, so a way that we could decouple maybe stress relaxation from stiffness, or we could try different peptides. We wanted to also try maybe different curvatures. So we were using this as primarily a means to study the corneal keratocyte. That being said, they could possibly find use in translation with implantation into a person's cornea. The reaction that we found was only light-mediated when there was a photoinitiator present. So I don't think that the reaction would take place unless there was a photoinitiator in the formulation.
So I guess you'd wash them extensively before you. Yeah, okay, great. And then I don't know if I have time for one more question. I was curious in chapter 5, you had this very nice calculation of the Gibbs free energy of your bonds of the associated. Okay.
Antonio Feliciano, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return in this room.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. South side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
applaus nemen.
Antonio Feliciano, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Dr. Baker is authorized to confirm upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Ready? Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I promise.
By the authority vested in us by law and in conformity with the decision of the committee here and present, I hereby confer upon you, Antonio Feliciano, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, the members of the committee, and affixed with the official seal of the university. I'm going to take a picture together.
Oh, nice.
Over here. Keep one hand under it. It's okay.
Give applaus.
All right, now I have to do the laudatio as well. First of all, thank you everybody for coming. Thanks also for the committee members, Rudy, Patricia, Ali, Stefan, whose name I do remember, Matt, Sylvia, and Lorenzo. I think you have a great team up here that supported you along the way, and a lot of us have seen you and worked with you in one way or another. So there's not much new that I can say today. I'll just give a brief summary. Tony, you joined our lab as one of the first PhD students and the first American candidate that we had. And I still remember your interview. We were talking and I said, do you really want to do this? And you say, yeah, I want to come over there. I want to do something new. I want to visit Europe. And some people now say that, this was a good choice. And I think that also for myself, that we both have experienced Europe in quite some interesting times. You walked into the lab full of energy, full of ideas. And I doubt you remember this, but during one of our first brainstorming sessions, we were talking about what we could do with the chemistry, where we could go, what interactions we could use, and you had listed 4 or 5 different supramolecular interactions. I said, look, it's a terrible idea to do your thesis on more than one supramolecular chemistry. You can see how well Tony listens. And I think that is a mainstay of your personality and of your character. A few people have worked with you in the past and work with you now. You really chart your own path. You're an extremely self-motivated individual, very creative, and I learned really quickly with you that sometimes the best way to encourage you was to get out of your way, to see what happens, and to help you puzzle through the tough times. You've been an incredible addition to the lab, not only from someone who has an eye for the details, but also an eye for the product, and how are we going to move this forward, and what are the challenges, and what are the limitations, but also with the personal aspect. So always being there for people, always ready to have a laugh, always ready to throw a Halloween party that I still never got invited to. I think that you have made a great career already, and you're going to continue along the path. I know that you've had an interesting industry career to start with, and now found a place that it looks like you're quite happy in. And I think that you have an extremely bright future ahead of you. And that's it. I have nothing else to say. Congratulations.
Thank you.
Dear Dr. Feliciano, also on behalf of the Board of Deans of the Maastricht University, I'm honored to congratulate you with this new degree and distinction. As already mentioned by your supervisor, work is going on also in research, and we will you all the best and a lot of success in your future career. Before closing, I will now close this academic ceremony. Before congratulating the new doctor, we will take a picture with the degree committee on the stairs in the central hall, and the audience can already go to the reception room. Hereby I close this academic ceremony. Thank you.
Dr. Feliciano. Wow, congratulations.